As the body runs through glycolysis, one thing we might find is that we're going to end up building an excess amount of energy. The body has a way that we can take that excess energy and actually run it back through glycolysis. In fact, even to a storage molecule called glycogen. That's what we're going to talk about today. Now, one thing we do not want to get confused here is that gluconeogenesis is not the building of fat. In fact, that is something entirely different. Today, what we're going to be talking about is actually taking pyruvate, our end product of glycolysis, and actually running that back through glycolysis in the opposite direction, and thus consuming ATP and consuming many of the metabolites that we made in order to produce a storage molecule called glycogen. So, gluconeogenesis is taking non-carbohydrate material and turning that back into a sugar, and what we do with that sugar eventually leads us to glycogen, which is a storage molecule. The issue that we actually face now, however, is that glycolysis actually had 3 committal steps where it involved ATP specifically in order to overcome a large free energy input. And so just how like the step 10 was a commitment step, we now have to use 2 enzymes in order to get over this one step. So this is what we're going to do. We're now going to use the enzyme pyruvate carboxylase with pyruvate in order to turn that into oxaloacetate. However, important about this mechanism is that 2 ATP are actually required in order to get it into oxaloacetate. Now you might remember, oxaloacetate is actually found in the citric acid cycle, and so it's kind of interesting to now find it in gluconeogenesis. Important to remember for later on when you're studying the citric acid cycle, oxaloacetate is actually found in a very, very, very low concentration in the mitochondria. And so because of this, you would not expect to find oxaloacetate going back up to gluconeogenesis. However, remember that gluconeogenesis actually happens in the cytosol and ER lumen. Okay, so back to oxaloacetate. So with our oxaloacetate, we're actually going to take 2 GTP with the enzyme phosphoenol pyruvate carboxykinase, and we're going to turn that into PEP, or phosphoenol pyruvate. Now with PEP, we're going to go ahead and run that through the same enzyme of enolase. And enolase is going to go ahead and turn that into 2-phosphoglycerate. And 2-phosphoglycerate is going to go ahead and use the same enzyme as before, phosphoglycerate kinase, which instead, this time is actually going to require ATP to go inside in order for us to get to 1,3-bisphosphate glycerate. This is an important step to remember. Instead of us getting ATP out, we are now putting ATP in. This is important. This is exactly why we run gluconeogenesis. With excess energy, we now need to store this energy for later. We don't need it now. So just like how our first 2 steps of gluconeogenesis are irreversible and are commitment steps, you could say, they also required energy. However, what's unique about this specific mechanism is that it actually inputs ATP. Instead, in glycolysis, you actually get ATP out, and that's what makes it reversible. And so from here, with 1,3-bisphosphate glycerate, we're going to go ahead and run that again using the enzyme glyceraldehyde 3-phosphate dehydrogenase. Instead, compared to glycolysis, we're actually going to use 2 NADH and 2 hydrogen protons and turn that into 2 NAD as well as 2 inorganic phosphates, leading us into glyceraldehyde 3-phosphate. Now in the presence of triose phosphate isomerase, we actually also get DAP, or dihydroxyacetone phosphate. However, mainly this reaction, just like glycolysis, only occurs with glyceraldehyde 3-phosphate. And so from glyceraldehyde 3-phosphate, we actually take 2 glyceraldehyde 3-phosphates and combine those together to form fructose 1,6-bisphosphate. And now we've hit the 3rd step of glycolysis, which is also another commitment step. And so to get around this, we need to use a whole new enzyme that we haven't used before, this being fructose 1,6-bisphosphatase. This reaction is actually going to require water to go in order for us to get an inorganic phosphate out. And that's very important considering what happens in the next step. Instead of going from fructose 1,6-phosphate, we're now going to fructose 6-phosphate, and so we're going to need that hydroxyl to replace the inorganic phosphate. Now from fructose 6-phosphate, we run it through the exact same enzyme as we did in glycolysis, again using phosphoglucose isomerase. No other reactants are needed for this one. Turn us into glucose 6-phosphate. And with glucose 6-phosphate, we now need a new enzyme to overcome a commitment step of glycolysis. And this time we're going to use glucose 6-phosphatase. And it's going to require, again, water to get out an inorganic phosphate. Again, going from a glucose 6-phosphate into a glucose. So no doubt we're going to need to take that phosphate out and as well as add a hydroxyl group in. And now here's a really crucial part about gluconeogenesis. We've come to the end point of gluconeogenesis of glucose, or even the starting point of glycolysis. What's important about this is that most muscles and brain and frankly most of the cells in the body are going to go ahead and run through glycolysis. And so with glucose now being dephosphorylated, it can actually leave the cell, because once glucose is phosphorylated and it turns into glucose 6-phosphate, or even later on in the liver, glucose 1-phosphate, it can no longer leave the cell with that charge. In fact, it won't be able to make it through those channels. And so now that's just glucose, we can shuttle it out into the bloodstream and send it to the liver. And that is gluconeogenesis. So in order for us to get to the end product of glycolysis, pyruvate, and turn that back into glucose, we needed to use 4 enzymes to overcome the 3 committal steps of glycolysis. It required us to use 6 ATP equivalents, as well as 2 NADH, and several waters. So while glycolysis was the catabolic pathway, gluconeogenesis is the anabolic pathway. We're going to go ahead and talk about glycogen synthesis in the liver next week.